This is a review for Exam 2 of Human Perception. And so we begin with the early visual brain, topic 8. So the first thing we talked about is where the visual information goes after it leaves the retina. So it travels through the optic nerve to the brain. 90% of the information goes to the lateral genicular nucleus, which, as we said, is part of the thalamus. The thalamus, as we'll see, is a way station that sends incoming sensory information to the cortex. But in addition, there are many other branches of visual information that go to other portions of the brain. So some of the information may go to a portion of the brain that is involved in eye movements, pupillary response, so dilating and contracting the pupil, sleep regulation, biological clock, circadian rhythm having to do with the pattern of dark and light. And so the idea here is that the visual information that enters the retina is kind of carved up and sent to these different locations to perform different tasks. We discussed the superior colliculus in relation to eye movements. And superior colliculus is a small portion of our brain, but it's the primary component, or actually the only component of certain non-mammalian species, such as frogs. And in those species, it's called a tectum. So it's a very similar structure. And it is involved in orienting movement. So when frogs turn left and right in response to prey or response to something that may be a threat, that kind of orienting movement is carried by the tectum. And in humans, we can think of this as being almost like our own amphibian brain, amphibian tectum, in that it's producing kind of simple responses in relation to the visual stimulation out there. So orienting to something that suddenly appears, something of interest, and also things like avoidance in relation to a looming object. These things may be carried by our superior colliculus. So, as we said, 90% of the information goes to the thalamus, the lateral genicular nucleus is just part of the thalamus, and so the lateral geniculate is one little portion of this much larger structure. And as we said, the thalamus is a way station for a lot of incoming information from different sensory modalities. So you have vision, audition, olfactory being the only exception, but tactile. All of these send information to the thalamus, which then relays that information to the cortex. And the cortex is, of course, the regions of the brain, this very large region that ends up doing all the high-level processing that we think of as really underlying perception. So the lateral geniculate, if you actually record from cells in the lateral geniculate, has properties that are in some ways similar to retinal ganglion cells. So they have center surround receptor fields, as we recall from the ganglion cells. It gets input from the eyes, but the lateral geniculate also gets input from the brainstem and other parts of the thalamus. So it's not only taking incoming information directly from the sensory input, but it's also coordinating with other parts of the brain, like the brainstem, which regulates simple physiological functions such as breathing, and other parts of the thalamus. So you'll get other modalities, other sensory modalities that which are represented in one part of the thalamus will then connect to the visual part of the thalamus. And it also gets massive feedback from the cortex itself. So it's really, as I said, it's kind of like Grand Central Station for all the information that's both incoming in from the sensory inputs and also traveling around in different portions of the brain. The lateral geniculate has a layered structure. There are 6 different layers. And 2 of those layers, the first 2, are what we call magnocellular, and the next 4 are parvocellular. And what we're seeing here is that there's a division of the visual input already from the point where it first is making its way into the brain. And so the parvocellular gets input from smaller ganglion cells. And these are, so the retinal ganglion cells, there are smaller ones that are sending information to these parvocellular layers, and there are magnocellular getting it from larger ganglion cells. And as you might expect, the parvocellular are providing input about fine detail and color information. So this is what we may think of as foveal kinds of functions. And magnocellular are taking information from more from the periphery in terms of motion and coarse form. So a major point that we see throughout the visual system is this division of labor that happens again and again with the same visual, with this visual input. In terms of pathways to the LGN, the thing to note is that information that comes into one portion of each eye, to the right side of each eye and the left side of each eye, and ends up in the same portion of the LGN. So over here we have the lateral geniculate nuclei, and these bulbs over here, and what you see is that the information that's going to the right side of the right eye, the right side of the retina on the right eye, which is of course coming from the left visual field, as we know, that information from the right side of the right eye goes to the right lateral geniculus, and the information from the left side, from the right side of the left eye also goes to the right lateral geniculus, and the same is true for the information on the left side of both eyes. So this leads to some information going to what we call the ipsilateral lateral geniculate, and some going to the contralateral. And so you have this crossing in the right eye. The information that's on the left side of the right eye is going to cross over and go to the left lateral geniculate nucleus. And what this does is preserves the it ensures that the information about a certain location in space, so information region over here, is going to end up getting processed in the same portion of the brain, rather than duplicating that information and processing it in two totally separate areas. As we mentioned, the layers, the 1 and 2 being magno, and 3, 4, 5, and 6 being parvo. If you look at the layers and how they're divided up, one half of those layers is going to be ipsilateral, and one half of those layers is going to be contralateral. So you have the 2 magnos being divided up, and then the 4 parvos as well. The lateral geniculate shows this property called retinotopic mapping, which is such that it preserves the spatial layout of the retina, which this means is that if 2 points are close together on the retina, then the receptive neurons in the lateral geniculate that are responding to those points in the retina are also going to be close to one another, and in fact they're going to follow in the same direction. So if 2 points are close together, say A and B in the retina, and B is to the left of A, then if you find those neurons that respond, whose receptive field is in those portions of the retina, so that here we have a portion of the lateral geniculate that's responding to this portion A in the retina, then when we move from right to left, we can encounter B, which is now responding to this portion of the retina that responds over here. So we have this retinotopic mapping, and it's going to be true across both eyes, and it's going to be true across all the different layers of lateral geniculate nucleus. So what this does is it makes sure that you kind of have a map. You can think of the brain as mapping the information of the light information that was first transduced on the retina. So what does this lateral geniculate actually do? Well, we know that it relays information to the cortex. The question is, why can't you just send them information directly to the cortex? So there's some evidence that it perhaps is involved in attentional modulation of input, perhaps it's emphasizing certain portions of the visual field. So you may want to emphasize what's over on the top left or on the bottom right, and the lateral geniculate may be involved in doing that. It helps organize the visual signal into various components, as we mentioned. There's the parvo and magno cellular layers, so it's organizing it, kind of filing it into different drawers. And it also integrates information from other modalities. So as we mentioned as well, that there are connections between different portions of the lateral geniculate, of the thalamus, to the lateral geniculate that are involved in other sensory modalities, such as hearing or tactile, and they can send information directly to the lateral geniculate. But as I said, for the most part, we don't actually know what the function of these are. All somewhat speculative in nature in terms of what exactly its role is. The next part of the brain, that the lateral geniculate, after it leaves these efferent projections out of the lateral geniculate, travel directly to the cortex, which is our outermost layer of the brain, evolutionarily newest, and in general is thought to perform most of the higher level functions in the human brain. And the first part, that portion that it reaches, is the primary visual cortex. Primary because it's first, and it's all the way in the back of the occipital lobe, and this is where the lateral geniculate nuclei send their efferent signals, and they project to this portion of the brain. as we noted in the relation to lateral geniculate, V1 also has a left side and a right side. And just like many other cortical structures, it's duplicated on both sides. But each side gets its input directly from the lateral geniculate on that side. So the left cortex, the left V1, and left, in fact, the entire left visual area is going to get its information from the left lateral geniculate, and the same goes for the right. What that means again is that the left brain is going to be getting information from both the left and right eyes, but the left brain is going to be getting its information from the right visual field, and the right brain is going to be getting its information from the left visual field. Once again, we have layers, in fact 6 layers in primary visual cortex, although layer 4 is further subdivided into 4 different layers. And these layers give it the title striate cortex, which means striped cortex. And these layers, similar to in the lateral geniculate, are such that they divide up the visual signal into different portions. And so we continue to see this division of the information throughout the brain. Again, in V1, just like LGN, we have retinotopic mapping, so that means things that are close together in the brain, in the cortex, so portions of the brain that are close together are going to be responsive to portions of the retina that are close together, as we see here. So if 7 is close to 8, so somewhere on here, over here, we have 7 and 8 on the contralateral side. So 7, and in this case, it's the visual field, and so it's 7 is above 8, and so 7 is going to be below 8 over here because of the way that the retina's information is flipped. And again, proximity here means proximity over here. And something else to note is that the fovea, which is a really small portion of the actual retina, is going to get a very large representation in the cortex, and this is called cortical magnification. So even though the fovea only makes up 0.01% of retinal area, it makes up 8 to 10% of cortical maps area. And what this means is that you can have much richer spatial representation. You can have much more information about that location, much denser sampling from the fovea, because you're now able to store and encode that information, and you have more cells to do it. And so you can think of it as sort of being, if you zoom into MapQuest, you can see the streets and the individual addresses, and so you have enough neurons to represent all the different information about this small portion. Whereas as you get further out in, on the retina, you have a much coarser representation, kind of as if you zoomed out of a map. A very important characteristic of V1 neurons, they demonstrate this orientation tuning. So they also have receptive fields, just like retinal ganglion cells and LGN neurons, which means there's a certain portion of the retina that drives the response of these V1 neurons. Some parts of the retina are going to make it go fire more, some are going to make it fire less. What you find is that they're elongated. These receptive fields are elongated, and they're kind of like sensor surround, but in this case, the sensor surround is such that it has a specific orientation. And so for a particular V1 neuron, there's going to be an orientation of a light signal that's going to make it fire more, and then orientations that are close to that are going to make it fire a little bit less. And then if you get really far away from that orientation, say a horizontal, in relation to a V1 neuron whose favorite response is vertical, you're going to see it fire even less. 3 types of V1 cells that we discussed were simple cells, which are the orientation of stimulus, as we just mentioned. Complex cells are responsive both to orientation, and they are also sensitive to motion direction. So as we saw in those videos that we watched in class, when you take an oriented bar and move in a certain direction, some cells will respond specifically to that motion. Another difference here is that simple cells' orientation is in a fixed location, whereas complex cells will often have orientation in variable locations. So you can have the same oriented bar, but in a different, somewhat different location on the retina, and it's going to respond to that as well. Hypercomplex or instap cells are just cells that, these are neurons that respond not only to orientation and even motion direction, but they respond to the length of the oriented stimulus. V1 has this columnar organization, and so it's a really remarkably structured organization such that you have these hypercolumns. So each hypercolumn contains a slab that responds to the left, primarily to the left eye, and another one that responds primarily to the right eye. And within each of these, you have these further subdivisions, such that as you move along within these columns, you get gradations in what orientation is the preferred orientation of those neurons. And as you can see, they're organized in a kind of fashion so that as you are closer together, you're closer together also in terms of how similar they are in orientation. And so this would be a whole hypercolumn, has both right and left in terms of which eye it's most responsive to, and they have all these orientations. And then if you move over here, you're going to get another one, which is actually going to correspond to a different portion of the retina. That's also going to have right and left, and it's also going to have all these different orientations. As far as the behavioral consequences of orientation tuning in V1, we actually can see behavioral consequences in the sense that there's different contrast sensitivity. So you need, contrast refers to how dark and light, what's the difference between the darkest and lightest points in an image. And so as we go from the top to from the left to the right, you see contrast is reduced. And what we know from careful experimentation is that certain orientations, such as vertical and horizontal, are more strongly represented in the brain, and therefore you don't need as strong a contrast in order to see them. And so if you do careful psychophysics, you can discover that we have better representation of vertical and horizontal orientations. We discussed the tilt after effect, where we had if you stare at a certain orientation, a grading of a certain orientation, in this case these bars of a vertical orientation, and then later on are presented with contrast thresholds for different orientations, you will find that you're going to get reduced sensitivity to whatever orientation you stared at for a long time. And this demonstrates that there are certain neurons that are specifically responsive to that specific orientation. So if you're staring at this for a while, basically the idea is those neurons get tired. The neurons whose job it is to encode this orientation, and therefore later on you're not going to have as good a sensitivity to this stimulus, and you're going to need more contrast in order to see it. Orientation selectivity develops, and the evidence for that is it's not something you're directly born with. It comes from kittens who were raised in the absence of, in this case, horizontal orientations. They lived in an environment where there were only vertical orientations, and what you see is that there will be no responsivity in their neurons or behaviorally to horizontally oriented edges. So this is a vertically reared cat here. It doesn't respond to horizontal edges, and a horizontally reared cat doesn't respond to vertical edges. And the idea is that if you're raised vertically, if the cat is raised vertically, it may see some orientations that are close by to this. So it's going to have some representation of things that are close to vertical, but as you get closer to horizontal, it has no representation of that. So what's the point of these oriented edges? One proposal is that this is really the basic building blocks of vision, that in order to encode things such as shape, complex shape, that allows you to identify things, one way to start is to piece together, almost like Tinker Toys, or you can think of them as a kind of individual Legos made in a chain. You can piece those together as oriented edges into any shape that you can think of. And this might be the basic means by which we first encoded visual information in order to identify it. Next topic was topic 9, visual brain 2. The question is, what happens after V1? So there's many different visual areas in the occipital lobe and beyond. And if we look at the mapping, so we have V1 and then V2 and then all these other areas that have been identified as being visual, and there's many connections between earlier areas and those other areas, feedback to these earlier visual areas, and so on and so forth. And so that's a lot of complicated connections. So what can we say about these? Well, we can summarize simply by pointing out that Parvo and Magno remain segregated. So, as we mentioned, you have Parvo and Magno in V1, which are getting, in lateral geniculate, which are getting their input from different retinal ganglion cells, and those are continue to be subdivided in V1, and then V2, which is the next section up. And then from V2, the parvo and magno continue to be segregated and sent to different portions of the brain. Magno ends up going to an area called MT, which is involved in motion perception, and parvo goes to an area called V4, which is complex shape. And so this division of the visual information continues into higher areas. Another thing we could say is that there's been a great deal of evidence for hierarchical organization. So if you record from individual neurons in the cortex, and as you get higher up in the cortex, the higher up you go, the more complex and the more specific the information that these neurons are going to respond to. So individual neurons in the retina, for instance, retinal ganglion cells respond to little spots of light, very simple characteristics. Just about any stimulus is going to be able to produce some sort of retinal ganglion response somewhere. If you then go to earlier cortex, the neurons are going to get more particular, a little more fussy about what they respond to, oriented edges. You get to middle cortex, so things like such as area V4, you're going to get responses to things like complex shape. So that's, you could think of this as being built of these more simple kind of units. And then if you take a bunch of these and to build an even more complex shape, you can get individual objects, things like faces and objects, and that's in the late cortex. So as we'll see, like areas IT, inferior temporal, you'll have neurons that respond to highly specific kinds of objects. Okay, so let's first talk about area V2, and for some ways it looks very similar to V1. You have these oriented responses to oriented stimuli. So you have receptive fields that respond to oriented edges, but it's also different from V1 in that it's responsive also to illusory contours such as these. So even though there's no edge in this location over here, you may see a V2 cell that responds as if there's an oriented edge there. Similarly, they respond to border ownership. So if you have a V2 neuron that maybe responds to this kind of dark light edge here in this orientation, it may respond when the edge belongs to the left, an object on the left, but not when that very same edge belongs to an object on the right. So how does this work? Well, V2 is doing something more complex than V1 neurons. They can't do things like border ownership or illusory contours, and likely depends on feedback from higher level areas that are getting information from lots of different portions of the retina, and then are able to convey information back to V2 that it should be responding. Area V3 is a big mystery. Of course, there's quite a bit of information about it, but it's not. We don't have a coherent story about what its function is. Area V4 is known to respond to complex shapes such as these. So as you, if you're recording from a single neuron in V4, you might find a neuron that responds maximally to a shape such as this one. And as you change shape systematically, you may find that it gets less responsive as you get further away from this particular shape. Even further along in the area inferior temporal, which is outside of the occipital cortex, it's already moved on to the temporal lobe, you find neurons that are responsive to highly specific kinds of stimuli. So this is recording from a neuron in inferior temporal of a monkey, and it responds specifically to faces, but does not respond to other kinds of objects. And in fact, in the hippocampus, there are neurons that have been found, let me just give it that respond to highly specific faces, or rather specific people. So there's a famous paper in which there was a patient who was undergoing open brain surgery, and they found neurons that responded specifically to Halle Berry. And when we say it responded to Halle Berry, what we mean is that it responded to the Halle Berry's face, also responded to a picture of her in a completely different setting, and even her name. And so this suggests perhaps that there are individual neurons that encode highly specific information about particular individuals. And this brings us to the a very basic question about neural encoding, which is how do neurons represent information in the world? So how does our brain ultimately represent the fact that there's a specific person standing in front of us, or a particular object? And so specificity encoding is the idea that individual neurons actually convey information about highly specific stimuli, sometimes called the grandmother cell. That it, the idea is that when a single neuron is firing, that neuron a specific neuron may actually signify that a specific, highly specific individual or object is out there in the world. Ensemble encoding suggests that that neurons don't carry such specific information, that it's not based on one neuron's response, but it'll be the pattern across multiple neurons. So, for instance, specificity encoding would say that there's going to be a single neuron that's going to represent the fact that you're looking at Bill or you're looking at Mary, and all the other neurons are going to be silent in relation to that stimulus. Whereas distributing or ensemble encoding suggests that many neurons may respond to Bill and to Mary, and what's going on at these levels is that it's the pattern across those various neurons that carries the information about whether it's Bill or Mary. And so in here you can see that all the neurons are responding somewhat to each of these different individuals, but none of them have the same pattern across these different neurons. Grandmother cells, as a hypothesis, has some severe limitations. One is that to be highly inefficient, you would need essentially a new neuron for every single specific type of instance of an object that you see in the world. Also, if we look at brain damage, we don't see people losing information about one specific object in the world or person, as would be expected if you could just lose that information by virtue of losing a single neuron. And it's also unlikely we would have found them. It's unlikely they would have found the Halle-Berry neuron if the Halle-Berry neuron really only responded to Halle-Berry and nothing else. We also did this in class tilt-after effect, where we looked at this adaptation stimulus and then and made this test stimulus look like it was oriented in the opposite direction. And we explained how this could be understood in terms of ensemble encoding, where if you look at the individual responses of these neurons here, so these are, what the representation here is that these are neurons that are fully charged up and they're responsive to these specific orientations. And so you're looking at this test stimulus, and what you see is this distribution of responses. So this middle one is responding the most because that's its favorite orientation, so it's responding, and these ones are responding a little less because it's not as close to its favorite orientation, and so on and so forth. And then after you adapt for a while, what happens here is this one here that responds to straight and is actually going to be weakened. It's not going to be responding as much as the one that's a little bit off to the left, that's favorite orientation is off to the left, because this one was oriented to the right. So this one that was oriented off to the left, it wasn't getting stimulated during adaptation. So now what you have is the ones that center and off to the right are weakened. And so what you get is a pattern of activation that's shifted over somewhat to the left. And so what you can see here is that, and that's the idea, is that it's meant to explain why you end up perceiving this as being oriented to the left. And it's really about the pattern, because any one of these, even this peak stimulation here, isn't really a very strong stimulation compared to, say, the responses of these guys over here before the adaptation. So it's really not about the fact that how strongly any particular neuron is responding, but rather the pattern over the entire array of different neurons.